Hello everyone, I welcome you back to this one-day workshop on virtual labs organized by GDC Ganderbal, Nodal Center for Virtual Labs recognized by IIT Bombay. And the first session will be conducted by me, Mudassir Roshan, Nodal Coordinator, and the technical session will be on life sciences. The list of the practicals which will be demonstrated, it is number first to determine pH of the wastewater, number second will be gel electrophoresis, and number third will be on light microscope. So let's begin the experiments on the portal of IIT Bombay. For this we have to go to Google and we have to type vlabs.itb.ac.in. If not this, you have to just type Virtual Labs IIT Bombay and you will be guided towards that portal. Now here we have to click on Virtual Labs IIT Bombay and will be taken to this portal. This is the Virtual Labs portal designed by IIT Bombay and it's completely free for all the users. And the broad disciplines are electronics and communications, which I already told you, computer sciences and engineering, electrical engineering, and then we have mechanical, chemical engineering, biotechnology and biomedical engineering, and then we have also physical sciences and chemical sciences. Today our session will be focused on that of the life sciences, so we have to go to this biotechnology and biomedical engineering lab. Let's go there, and here you will be guided towards these practicals which are available in this biotechnology and biomedical engineering domain. Here you will find variety of labs. It will be on bioinformatics, system biology, computer aided drug design, ecology, biological image processing, bio-inspired robotics. But first of all, we will go to ecology virtual lab. Click on it and you will be directed here. In this ecology virtual lab, it is designed by Amrita Vishwavidyapeetham University. And here you can find n number of practicals related to ecology, which are relevant to both UG and PG, and even to research, determination of pH of wastewater sample, biological oxygen demand, chemical oxygen demand, nitrogen cycle, introduction to species interaction, and so on and so forth. Our first practical will be on determination of pH of wastewater. So let's click on it and it will take us to the experiment. Here you can see there is theory, procedure, self-evaluation, animation, assignment, reference, and feedback. In the theory, there you will get complete backdrop of the experiment, why we are performing it, what is the need, what is the principle behind everything. Then we have the procedure. In the procedure, you will be guided towards what are the materials required, what is the procedure, and then in self-evaluation it will evaluate you, it will help you to know what you have learned from it, and then in the animation it will guide you towards the step by towards each and every step of the experiment by visual aids, and then you have the assignment, reference, and feedback. First let's go to theory. In the theory, as I already told you, it will tell you what is the purpose of the experiment. See, if we will see determination of pH of wastewater sample, firstly we have to know what is pH. pH, we know it is the concentration of the hydrogen ions, and the pH of the solution, it is rated 0 to 14. It can be 0 to 14, and it can be acidic or basic. In our day-to-day life, it's important to know the pH of the water for its purity and so on and so forth. Also in the labs, we want to know the pH of the buffer solutions, so on and so forth. So let's see how the pH is determined. Now we have to go to procedure. In the procedure, it will tell you what are the materials required. You require wastewater sample of the water sample of which the pH is to be taken. Then you require acetate buffer. Acetate buffer is pH 4, means it is acidic in nature. Then we have ammonium. pH is 10, means it will be alkaline or basic. This knowledge you will be having. Then we have the pH meter, and then we require the tissue paper. Then it will tell you what is the procedure. Okay, you have to collect the water, you have to switch on the pH meter. Step by step procedure will be given to you. So let's go to the animation. In this animation, you can see how the pH of the wastewater or any sample of the solution is measured. In these times of pandemic where you can't go anywhere, it's very relevant to use these virtual labs from your home, where you can get the basic ideas about what virtual labs are, what are the instruments, how they work, what are the steps. It will guide you completely. So let's see. This is the step which will you will require for this virtual labs. You have to press start here. See, one thing more, before using this portal, if you are the first time user, you have to sign up free. You have to create an account. If you are a user already, you have to log in. And the next thing is, to use these virtual labs from IIT Bombay, you need Adobe Flash Player on your PC, on your mobile phone, and you have to first download it from the Play Store, and then you have to install it, and then you have to allow the Google Chrome or any web browser you are using. Okay, now let's come back to our experiment. It is determination of pH of wastewater sample. This is the setup you require for determining the pH. See, this is the pH meter and it's connected to the electrode. Okay, this will give you the reading of the pH and it's connected to this electrode, and this electrode, it's always stored storage solution which is 4 molar potassium chloride. If 4 molar potassium chloride solution is not available with you, you can store it in acetate buffer of pH 4 or ammonium buffer of pH 10, but never store it in deionized water or distilled water, for it can damage the glass electrode. So we always, whenever the pH meter is not in use, we do store this glass electrode in the 4 molar KCl solution, which we call as storage solution. Okay, then we have this acetate buffer. This solution, this is the having pH 4, means this is the solution of known pH 4, acetate buffer, acidic pH. Then we have the ammonium buffer, that is pH 10. Again, this is a solution of known pH. Then we have the waste beaker. This is labeled as waste beaker here. Here you can put, rinse the electrode and put the discarded distilled water solutions, it's straw here. Then you have the tissue paper which you use to wipe the electrode, and then you have the sample of the water of which pH is to be determined, and then you have the distilled water here. Hope everything is clear here. Let's continue. Now, firstly, you have to take the water sample of which pH is to be determined. It can be even any solution. You have to pour the water in wastewater sample. Then you have to turn on the pH meter, and then you have to take the glass electrode and put it in. And then you have to rinse it so that no storage solution is attached to it in the wastewater. Then next you have to take the tissue paper and wipe it. Then you have to dip it in acetate buffer. Glass electrode is next, dip it in this acetate buffer of pH solution 4. Then this is called a standardization. Here this is known solution, you know the pH is 4, and your pH meter should also show that the pH of this solution is 4. If it is not 4, you have to use the calibration button to put the pH, to put this reading to 4. This is called a calibration. Similarly, now after this acetate buffer calibration is done, you will again rinse it with the distilled water and again take a tissue paper to wipe it. And next you have to do the standardization in the ammonium buffer solution of pH 10. Firstly, you will rinse it. Again, take a tissue paper, wipe it. And now we will go to ammonium buffer solution pH 10. Again, we know the pH of the solution is 10 and the meter should show us the 10 pH. See, this pH meter is showing the 10 pH, so we don't need to do anything. But in case it will not, we have to manually calibrate by using the calibration button. We have to put it on 10. This is called the calibration or standardization of the pH meter. After the standardization is done, now you can again rinse it with distilled water. Each time you have to be careful about rinsing. When you switch on from one solution to another, you have to rinse it with distilled water, otherwise your measurements will not be accurate. And then into the storage solution, and now again rinse it. Finally, after the calibration, proper calibration is done, and we know that the pH meter is showing the accurate readings with the known solutions. Now we can take the pH of the unknown solution, and it is showing 5.50. Again, so we are done. We have taken the readings of the unknown sample as well. We have to put it in back in, we have to rinse it and put it back into the storage solution. The reading is displayed as 7 in the storage solution. This is all about this experiment of this determination of pH of wastewater sample. Now after going to this, you have again one more option, that is self-evaluation. You can go there. In the self-evaluation, you will find the objectives. When you will which of the following best describes pH? It's the measurement of the hydrogen ion activity, so on and so forth. You have to see, and then at the bottom you will have to click submit. It will show you that what's your level of understanding, and then you have the assignment, home take, and then you have the feedback. With this, our first experiment is complete. Now let's go to second one, that is agarose gel electrophoresis, that you will find in molecular biology lab, virtual lab 1. Here also you are having n number of practicals. Click on to the one you want to do. It's agarose electrophoresis. Again, like the same way, you will be having theory, you will be having procedure, you will be having self-evaluation, animation, etc. Agarose electrophoresis, if we want to know, it's the easiest and most popular way of separating and analyzing DNA. If you have say for example isolated DNA from a sample, and you want to see whether the DNA is present there or not, or you have done the PCR and you want to know whether the DNA is there or not, you have to go for agarose gel electrophoresis, which is the easiest and the most popular way of separating and analyzing DNA. So let's go to the procedure. This procedure will show you exactly what you require, which are the buffers and solutions you require, what are the instruments, what are the equipments. It will guide you thoroughly through it. And now then will be self-evaluation, and then we have the animation. Let's go to the animation quickly. Again, you need Adobe Flash Player for this virtual lab to run animations. Now this is the setup for this agarose gel electrophoresis. Let me introduce you to this equipments used. This is the microwave, which we also use in our common homes, and this is the gel electrophoresis unit. This is its voltage stabilizer, and this is the gel tank. And then you have the gel casting trays in which you pour the agarose gel to set, and then this is the micro pipette, and then this is distilled water conical flask. These are the beakers. This is the measuring cylinder. This is UV illuminator on which you see the DNA finally, and this is the weighing balance. This is all you require for this agarose gel electrophoresis. Let's continue. Firstly, this is the micropipette. You require 2 ml. It's a mistake here. It's not 2 microliter. You require 2 ml. 2 ml is equal to 2000 microliter. So this micropipette will support from 200 to 2000 microliters, and you have to set it to 2 ml. 2 ml means 2000 microliters. So let's set it to 2000 microliters. And then you have to take 2 ml of TA buffer stock solution, tris acetate EDTA. This is on the virtual labs. You can see how this TAE is prepared. Now imagine we have the TAE buffer stock solution which is already prepared, and we have taken 2 ml from it and are transferring it to 100 ml of measuring cylinder. So we are having 2 ml of TAE buffer which we are putting in this 100 ml measuring cylinder. This is the 50x buffer solution. You can see in another practicals on this virtual lab how this TAE buffer is produced. Take 2 ml from it and put it in 100 ml of measuring cylinder. Next, take 98 ml of distilled water to make this 100 ml. Now put this buffer in a conical flask. This is 2 ml of 50x TAE buffer plus 98 ml of distilled water. Total measurement is 100 ml. Now take agarose. Now what this agarose is, it's tin, it's a polysaccharide and is extracted from certain red algae. It's frequently used in molecular biology for separation of large molecules, especially DNA. It's available in white powder. Well, the more the concentration of agarose we put in the solution, it will prevent the movement of large DNA molecules, means the pores will be small. Okay, when we go on increasing the concentration of this agarose, it will be dense, the solution formed will be dense and the pores will be smaller, so it will hinder the movement of large DNA molecules. For the movement of large DNA molecules, we have to reduce the agarose amount. For now, we are weighing 2 grams of agarose to make 2% of agarose gel. We have kept the buffer up to 100 ml. Now we will put 2 grams of agarose into that to make 2%. Means 2 grams in 100 ml is 2%. So let's weigh the agarose on the weighing balance. This is the spatula which is used, and here it will show you. Always remember, you have to calibrate it to 0. You have to put the measuring balance on 0, and this is 2 grams now. You have to take this agar and put it in the solution. Initially in the coldest solution it will be insoluble. It will not solubilize. For this you have to put it in the microwave. You have to boil it so that it will solubilize in it, and you have to put the microwave on 85 degrees Celsius and let it to start for 2 minutes. For 2 minutes you have to put it there. After 2 minutes you will see agarose would have completely dissolved in the buffer, TAE buffer. And now after this you have to cool it, so that you cool to an extent so that you will be able to touch it. Okay, now you have to again take a micro pipette, this time from 0.5 to 10, and you have to adjust it to 0.5 microliters. You have to take ATBR. ATBR is ethylene bromide. It's an intercalating agent commonly used as fluorescent tag, and abbreviated as ETBR. When exposed to ultraviolet light, it will fluoresce with an orange color, intensifying almost 20 folds after binding into DNA. Means this ETBR, it will under UV light, it will give orange color. So this ETBR will show us that whether the DNA is present there or not. It is an intercalating agent and it binds to DNA, and when you will get an orange band under the UV light, it will show that the DNA is present. But you have to be careful with this ETBR because it's a carcinogen, it's a potent mutagen, and so you have to use it carefully wearing gloves. Let's point 0.5 microliters of it with the micro pipette. put it in the solution in which we have agarose and TAE buffer. It's still boiling and we have to dissolve it, shake it with the hand. Then you have to take the gel caster. This is a gel tray and you have to put the agarose in it to set. You have to tighten it. These are gel casting trays. They come in a variety of sizes and are composed of UV transparent plastic. Now you have to place this. It's called as comb. For this comb will give you the gels. Now you have to pour it in this agarose solution. It's liquid, but when it will cool, it will set. Allow the agarose gel to set. Now, after it has set, remove the comb carefully. You will see the wells have been formed. And take the gel tray out. Now this is an electrophoresis chamber or tank and you have to put this tray along with the agarose gel slab which is formed into it, and you have to put again TAE buffer in this gel tank over it so that all the wells are completely dipped in it. Now you have to take the parafilm, or the in place of parafilm you can also take the Eppendorf tubes, separate Eppendorf tubes. And now you have to take the micropipette, adjust it to 0.4 microliters to load the dye. This is loading dye. Loading dye is generally bromophenol with glycerol. You have to take 4 microliters of the loading dye on a parafilm. You have to spot it for as many as DNA samples you have. One DNA sample will go in one well. Remember 4 microliters, but you can adjust this slightly. It can be 2 microliters even. This is just dye. And here is taken 6 spots. Now you have to adjust it to 20 microliters. Now again, it depends upon the amount of the sample you want to load. You have to take the DNA sample which you have already extracted. It depends whether you want to load 10 microliters, 20 microliters accordingly, but here it's showing that here it's shown that we have to take 20 microliters. So let's see. Firstly, it is the marker or the ladder which is taken. These are the tips. And 20 microliters have been sucked into this. And now we will mix it with this loading dye. Now we have 20 of this and 4 of the dye, and we will adjust this microliter to 20, this micropipette to 24 microliters. 24 microliters we have to adjust it. We have to press the button to suck in, and then we have to put it in the well. This is the well over here. Without any water bubbles, we have to pour it in one go. We don't have to suck it back, otherwise the solution will come out. It's just in one go, and this process called as loading. Again, we have to adjust it first to 20. Take the DNA sample. Now sample 1. In this, we have to check whether the DNA is there or not. Discard the tip. Each time you switch on between the samples, you have to discard the tips. Take 20 microliters. Again, mix it with the loading dye. This process is done so that the sample and the loading dye are mixed properly. And now adjust the volume up to 24. 4 of dye, 20 of the sample. Now we have to push the micropipette. We have already adjusted it to 24, and now this will go in another well. This is called as loading. again we have to go it in one. We don't have to pull it back, because that will take the sample out. Like this, we have to load each and every sample in the separate wells. But each time we load different samples, we have to discard this tip. Now we have to put this in this gel tank. We have to cover it and then connect it with this voltage. We have to give the voltage to it. And remember, these wells are to be kept on the negative side. Now the voltage given is 100, and now these wells are on negative side, and DNA will move from negative point to positive, because DNA is itself negatively charged, so it will be attracted towards the positive. When the 3/4 of this bromophenol dye it has passed of the gel, you have to switch it off. So that it won't run out of this agarose gel. Now see, you have seen that these blue lines have moved, but it doesn't mean the DNA is there. It is just the loading dye which is blue in color. Now to see whether the DNA is there or not, we have to transfer this agarose gel slab to the UV illuminator, and we have to on the UV light, and it will give orange bands. See, this is the marker or the ladder. This will show you the KB size of the DNA fragments, and this is DNA present here, DNA here, DNA here. This will give the DNA fragments. This is all about gel electrophoresis animation. Now also they have given you the simulator here. Let's load on the simulator. Now let us go to the simulator of this gel electrophoresis. After going through the procedure, we now know what to do. See, here you have the variables which you can change, and accordingly you will get the results. Now firstly you have to select the concentration of the gel. In your this animation there was 2% of agarose gel, but here we will be using like 0.7. It is like higher the concentration of the agarose, lower DNA molecules of lower size will travel, and the larger molecules of DNA won't be able to travel through that, and vice versa. Now let's select the DNA. Let it be PBr322. Let's set the marker up to say this 6 kb. The marker you have to adjust according to the concentration of the agarose you are using. And this PBr322, it's our DNA sample, and let us say in well 1 it will be acted upon by the EcoR1 restriction enzyme, and we want to see what will be the results. Likewise in second well we will be again having the plasmid PBr322, but this time we will select the enzyme to act upon it is EcoR2, and likewise we can see what will happen to this PBr322 by another enzyme, so on and so forth. By now we are selecting 2 enzymes to see what will happen to this PBr322, what digest results we will get. So let's start the simulator. Now this is the marker, and here this will be the well 1, wherein this PBr322 will be acted upon by EcoR1 restriction enzyme, and we will see what DNA we will get, what digest products we will get. And in the second well, same PBr322 will be acted upon by EcoR2 restriction enzyme. So let's turn the power on of the gel electrophoresis. Here we are applying the voltage. The speed of these DNA molecules will also be determined by the voltage we have. More the voltage, more will be the speed, but we have to be cautious that we don't apply so much voltage that it will heat up the agarose gel and it will melt. We have to be careful. Usually it's 110 of voltage that is applied normally, and we have to wait till this bromophenol which is electrophoretic marker travels 3/4 of this slab, and upon that we have to turn off the power. Otherwise these DNA samples they can run out of this agarose slab. And remember, it is like negative electrode of the gel electrophoresis, and this is positive. And DNA is always loaded at the negative because DNA itself is also negatively charged, and it is attracted by the positive pole. That is the theory behind this traveling of DNA. Means it gets repelled by the negative pole because it also has charge negative, negative, negative repel, and it just gets attracted towards the positive pole. So let's assume that these have run and we turn off the power. Now these bands don't mean that it's DNA, it's just electrophoretic marker which shows that the samples are running. Now to visualize the DNA, we have to turn on the UV, and it is the ETBR which we are initially adding when we have to make the gel slab, which chelates with the DNA and makes it visible. So let's turn on UV. Here in this you can say this is a marker, and in the well one we have just got one band of DNA, and in third we have this eco R second acting upon PBr322, and we have 3 DNA bands. Hope this is clear. Now let's move to another experiment, that is light microscope. Light microscope, its binoculars is a version of light microscope. Let us see how it works. Light microscope we use for visualizing of stomata, we see these algae, bacteria, so on and so forth with that. So let's see how this binoculars is operated. We are having just two things here, one is simulator and another is the animation. So let's see what's the animation. Click here. It will take you to the animation. It's a YouTube link which beautifully explains how to use this binocular microscope. It is loading. Let's wait for some time.
Light microscope. The different parts of the microscope are eyepiece, coarse focus knob, switch for light source, brightness control, condenser, etc. Switch on the light source. The switch is placed at the base of the microscope. Adjust the light intensity with the help of a lamp rheostat. Place a microscope slide on the stage. Rotate the nose piece to the lowest power objective, that is 4x objective lens. Observe through the microscope and move the iris diaphragm lever till most of the light comes through or the background becomes brighter. Close 3/4 of the iris diaphragm. Adjust the coarse adjustment knob until the specimen is in focus. Rotate the fine adjustment knob till the image is becoming clearer with good resolution. Rotate the nose piece to the 10x objective for 100x magnification. Open half the iris diaphragm. Adjust the coarse adjustment knob until the specimen is in focus. Rotate the fine adjustment knob till the image is becoming clearer with good resolution. Rotate the nose piece to 40x objective. Open 3/4 of the iris diaphragm. Focus the lens using coarse adjustment knob. Rotate the fine adjustment knob until the image becomes clearer with good resolution. Rotate the nose piece such that the specimen is between 40x and 100x objective. Place a small drop of oil on the slide in the center of the lighted area. Rotate the nose piece so that the 100x oil immersion objective touches the oil. Open the iris diaphragm completely. Only fine adjustment is done. The image will become clear. Remove the slide and wipe the 100x oil immersion objective carefully with lens paper to remove all oil.
Well, this is all about this light microscope of binoculars, usage of binoculars. Now let's go back. It has a simulator as well. Let's click on it and click on the simulator and see how we can use it. Here we are having the monitor out. Let's choose binocular, and then we are having 10x. You know, the magnification power, it's 10x for takes 100x. Let's assume that it's 10x, and let's assume that our sample is unstained onion epidermal cell. Now we want to visualize it. See, this is power on and off. This is light on and off. You know, we have to adjust the light, and this is the light intensity up and down, and this is by this we can move the view of this towards up and down by clicking on these arrows, and then this will move it to right, and this will move it to left, and then we can press here to zoom. You can see it zooming, and then to zoom out you have to again press this. Okay, then we can see how it will look at 40x. We'll magnify it and we'll see how it will look at 40x. This is the picture of the onion epidermal cells, which are somewhat rectangular in shape, and let's zoom it at 40 magnification. The epidermal cells of the onion will look like this, and now if we'll go for 100, they will further magnify. These are the samples. For example, we want to now, instead of unstained onion epidermal cells, we have gram negative rods. We can change, and at 100x they will look like this. Then another sample can be cocci, gram negative cocci. They will look like this. Then we can also see how bacillus subtilis looks, rod-like structure. This is how it looks. This is all about this usage of the virtual labs from IIT Bombay. Hope it's clear. We are looking for your feedback. Please give feedback on the portal of IIT Bombay. The experiments done are determination of pH of water sample, agar gel electrophoresis, light microscope. This is in life science. It's demonstrated by Mudassir Roshan. Lab is Amrita Vishwavidya Peetham, Nodal Center number 208, GDC Gandarbal. Remember, you need this to provide the feedback to us on the portal of IIT Bombay. I thank you all for joining us for this workshop. We'll be conducting more workshops in the near future on life sciences again, physical sciences and chemical sciences, and of course computer sciences. Now the second technical session will be led by Professor Asya Koyum, pertinently whose research is also on the simulation. She's the technical coordinator of this as well. Thank you IIT Bombay for providing us the usage to these virtual labs, wonderful virtual labs. Thank you GDC Gandarbal for organizing this workshop. Thank you all.